Hi, everyone. Thanks for joining us, and welcome to the latest episode in our new webinar series, Oncology Hour. I'm Amanda Holman, and joining me this week is Dr. Arturo Loaiza-Bonilla, our Chief Medical Officer and practicing oncologist. We work with Massive Bio, a concierge clinical trial enrollment service where we connect patients to clinical trials. We're here to discuss cancer and any questions you want to know. We know there are always lots of questions surrounding genomic testing. So for this week, we'll be discussing how genomic testing is used for cancer, and based on community input, we've created a Q&A. If you have any genomic testing questions that come up, please write them in the chat and we'll make sure to take time to answer them as we go. Before we begin, Dr. Bonilla, can you tell us a bit more about yourself?
Sure, absolutely. Thank you so much, Amanda, for having me today. So I'm a practicing oncologist, born and raised in Colombia, came to the United States, wanted to do research, and I became passionate about oncology because of a family member who actually had cancer, and started to look into biomarkers and ways to improve the access to patients to clinical trials, looking at testing as a way to do that. The way of the future these days is by looking at the genetic makeup of the cancer itself and try to match the patients to the best options, including clinical trials, use technology, et cetera. So I've been around seeing patients over a decade now. My main focus is GI malignancies, but anything related to precision oncology or focusing on genomic markers is my passion. So thank you so much for having me, and hopefully we can answer some good questions from the audience.
And we're really excited to have you here. I think the very first question is, can you explain in a bit more detail just what a biomarker is?
Yeah, absolutely. So let me just see if I can share my screen for a minute, because I knew I was prepared somewhat for this. So let me just do this. Okay. So basically, when we talk about biomarkers, we are focusing on cancer specifically. We know is a very relevant disease that is the leading cause of death worldwide, and we need to find options for patients. So in the past, we used standard approaches for all patients. So before personalized medicine, the same location and stage of a cancer required the same treatment, right? So we didn't change anything. We say it was coming from the breast or the lung. We treated the same way, regardless of any other characteristic of the tumor, at least in big picture. But as we started to decode the DNA and the proteins that are involved in cancer and in other normal processes and abnormal processes, then we started to look at those biomarkers. So first, just to set the stage, so what are genes or proteins? So genes is basically the book that the DNA contains. So basically, the DNA is the book, and you open the book, and it has genes. Those genes have coded messages that tell the cells how to behave. So you want a recipe for a cake, open the book on page number 23, and you're gonna have the recipe there. So that's basically the information that you need. Proteins carry out the messages from the gene. There's an intermediate called RNA, which is becoming very relevant because of the vaccines and all that. Basically, the RNA is the translation of the book, and then the proteins are the final product of that. When cancer develops, because there's a genetic mutation that gives you a typo, a problem on the book, and that leads to a cake tasting salty instead of sweet, or a problem that of course this is an analogy, but that's how cancer develops, right? The cell is not having the right manual to perform its functions. So what is a biomarker? A biomarker is a molecule that you find in blood, other bodily fluids, or tissues, and it's a sign of a normal or a normal process. So you can have normal biomarkers, right? So the things like, for example, checking a molecule in blood, for example, cholesterol, and being normal, that is a normal process. And then you find is when that number goes in a different direction, that you start looking into it in a much more specific way. So biomarker test results can help match patients to a cancer treatment if we find them. For example, we find abnormalities in the DNA in the bloodstream. And that basically leads to the concept of precision oncology, which is basically every single cancer has different proteins and different gene mutations. And if we can target what is driving the cancer growth, then we can potentially use that biomarker to attack the cancer itself. So with that, I'll just hold it for now, but that's kind of like introduction of biomarkers.
Thank you. That's really helpful and really interesting to learn as well about how you can specifically target a biomarker to treat a cancer in such a personalized way.
Absolutely.
So we actually, before we move on to our next question, we have a community question asking, how can a cancer patient get genomic testing?
Okay, so genomic testing is one of the multiple ways to look at the cancer biomarkers. So let me see if I have a slide here that I can actually show you some of this. Okay, so there are different biomarker technologies. One is the typical ones, which is called immunohistochemistry, which is using basically stains that you use like inks that you put on the tissue itself that is cut and fixed on paraffin. And you look at under the microscope with different, biomarker staining tools, and it shows you things that are there in a higher or lower expression. The other one is called FISH. So FISH is a biomarker technique used to detect a single gene DNA alteration. So in this case here, we can see the different genes are marked with different colors in a fluorescent light. And you can see when things are fused or change because of the change in color and in configuration on these cells. When you talk about next generation sequencing, that's what we call comprehensive genomic testing. It's basically looking at, instead of looking at a single gene or a small panel, we're looking at the whole area of interest in the book. It could be what we call a whole exome sequencing, basically looking at all the readable parts of the book, go through all the pages and look for the typos to see what's going on with the recipe. Or we can do hotspots where we go and say, okay, I want to know everything that is related to, the ingredients of the cake. So in page 10, I know there's one ingredient that is involved in the cake, and then in page 25, there's another one. Let's look for typos in those different pages about different genes. And the bigger the number of genes that are involved, the more comprehensive the panel becomes. So it's basically looking at comprehensive genomic profile by doing that. And that's how you get it done. The way that can be done is typically by taking a piece of the cancer, looking at the DNA structure that is there, like if there's enough DNA in those samples, it gets sent out for the testing, and we do that process. So what we call it next generation sequencing. So what is new about it? So this is something from Jen Morris that I borrowed. Basically, there's two ways, for example, to do this comprehensive genomic testing. One of them is looking at changes on the DNA is comprised of different letters that are basically base pairs, and they have different pH. They're more basic or more acidic. Basically, they have a different pH that a machine can detect based on those changes. You can really tell what are the base pairs that you're reading. So you can see the signal for, different things happening there. And the other way to do it is based on tagging, which is when a base is added, a fluorescent signal is detected. And that's another way to detect those genes specifically in those mutations in different places based on tags that you add on into the PCR process. So so that's how you do it. There's other way to do it, which is doing it in the tissue itself, as I mentioned, or you can do it in the blood, and that's called a liquid biopsy. So a liquid biopsy, it's basically looking for those tiny pieces of DNA that are released in the bloodstream by normal processes as the cancer cell is dying and releasing those, DNA that was inside of them. You can tag in the bloodstream those mutations as well and find through a test, like a blood sample, those mutations as well, almost at the same level as doing a tissue-based biopsy, at least for the genomics component. Like you're looking for genes, then you can do it. For protein expression and others, you may need the tissue. But for what we call comprehensive genomic panel, we can right now use liquid biopsies and we can use tissue biopsies. So both of them are available for our patients, and you just have to talk to your doctor because it's something that is currently reimbursable for most patients and it's available. and it can help you find clinical trials and optimize, hopefully, your care. If you have a biomarker, you may get benefit from a different drug that is tailored to your cancer.
Thank you. What are the benefits of using like a liquid biopsy versus a tissue biopsy and the different types?
Right. So I think in terms of liquid biopsies, the potential benefits that are being described are, first, the turnaround time. So you can get the liquid biopsy results sometimes in a week. So let's say you see me in clinic, I get discuss with you. It's like, well, you're coming for a second opinion. Your tissue is still 25 miles away or more in another practice or another laboratory. And instead of me requesting it, getting the sample, getting to see me, and send that test out for tissue, or even order a new biopsy, which is time consuming. Now you simply draw the blood at the same time in the office, and then send it out. So it's quicker. You get all the same amount of information, typically, that the typical comprehensive genomic panel testing, and you can also find kind of like the heterogeneity. Cancers, sometimes they have different areas, even between the same tumor or across the when the cancer is advanced. Sometimes there's what we call clonal and subclonal mutations. Basically, not all cells in the cancer are equal, so they may have a different clone, and that means that you may not be representative of a single biopsy. You do put the needle in a place. That gives you a pretty decent idea of the cancer itself. Don't take me wrong, it's like a very good testing. But also the blood liquid biopsy assay can give you maybe some subclonal populations that were not detected, because it's telling you the whole story of the whole body, right? As long as there's enough DNA from the tumor in the blood shedding that you can actually capture. So those are the preferred, like, kind of turnaround time and detecting more things quickly when tissue is finding. So in lung cancer is a no-brainer. We know that doing liquid biopsy or plasma first approaches is the way to go. So, and we really need to test every single patient with non-small cell lung cancer in that stage with a comprehensive genomic panel, because there's so many biomarkers now that you can benefit from, that certainly is warranted to do it. And not having a comprehensive panel for these patients it's probably not the best approach. So I absolutely recommend those patients to get liquid biopsies. If they want to find the rare interact fusion or something like that, then you can also order the tissue-based assay, maybe as a sequential approach, or concomitant. It's up to you, but certainly you need to get something done quickly.
Along those lines, are there certain biomarkers that are commonly seen across cancer types?
Yes, so let me just see if there's anything here. So interesting enough, over the past 5 years, we had so many new indications for every single kind of cancer that our minds are blown. Like, we need help to really even keep up with all these things, with all the new biomarkers, the new drugs coming, the 700 new molecules coming as well in the pipeline. But just to give you an example, this is non-small cell lung cancer. And just look across how many biomarkers are present that can be driving the cancer itself, right? So this is from the TCGA data. So 75% of lung adenocarcinoma drivers are known. That means that we can target them with different drugs. Now we have 8 FDA-approved drugs for lung cancer, for example, that are targetable, including a new one called KRAS G12C, which was undruggable until recently. Now we have opportunities for that. So, but what we learned is that there's sometimes ways to attack the cancers in a tumor agnostic fashion. That means that instead of looking at the tumor itself in terms of location, sometimes when they share similar genomic or protein-based characteristics, we can target those tumors in the same fashion, regardless of the origin, right? So this is just a list that is not comprehensive, but is pretty robust, telling you what are the most common actionable biomarkers, either by next-generation sequencing or immunohistochemistry across the board. You can see this is colon, lung, breast, cholangiocarcinoma, prostate, pancreas, gastric. So it covers pretty much the vast majority of cancers, not even including the hematological, which is also another story. But there's a number of genes that you can start seeing that are commonly shared across different tumor types. If we find those biomarkers present in next generation sequencing, we can potentially help patients to be enrolled in either clinical trials, or we can give a patient a drug that is already FDA approved for the indication. So really helpful to get genomic testing, because it's really going to help you to find the best options for your care. And that's exactly what I want for every one of my patients. I work at Cancer Treatment Centers of America. We're very proud of being the very prolific on doing genomic testing and helping our patients. So once again, I think that's something that we should recommend in all of our advanced patients, just to find those options, because if you have those biomarkers, you're really going to probably help the patient in a much more efficient way.
So I think I'm going to know your answer, but based on what we just talked about, would you say that any patient who's been diagnosed with cancer should get genomic testing as a best practice?
Yes, but with a nuance, I would say genomic testing for therapeutic purposes should always be for every advanced patient. Basically, a cancer that has gone beyond the site of origin, that it has gone elsewhere. Absolutely, 100%. All my patients should be tested for that. There's other opportunities for doing genomic testing. So there's, just to make a differentiation, there's genetic testing and genomic testing. What we call genomic testing is basically testing the tumor for alterations, right? Which is what we were just talking about. There's also genetic testing, which means that you talk to a genetic counselor and you check for potential genetic abnormalities in your body that you were born with, right? So that's the genetic component of that. Also recommended for a number of tumor types, because it can help you to guide treatment and help you to guide your family, as well as if they can have any other family members with cancer. But the short answer to this is that yes, any advanced cancer patient should have benefit of getting genomic testing at least once. If you are on targeted drug that targets specific mutation that you were being treated for example, lung cancer, you have an EGFR mutation, and you take a drug for that that is controlling your cancer, and then you have progression of disease, then is warranted to potentially do another biopsy or liquid biopsy, because those that are being treated with prior targeted drugs can develop new mutations or have passenger mutations taken over, and it makes sense to repeat it, because that's what we call as clonal evolution. So not all cancer types are like that, so talk to your doctor, but at least once you should have that, right? So it's something that is a call to action, because we don't see that enough, even in diseases such as lung cancer, that are very well known to have so many biomarkers, that is absolutely cost effective to get genetic testing done for those patients, genomic testing as a whole. So that's the message.
Thank you. I was going to ask about the difference between those two, so it's very helpful to explain. What are targeted therapies? I know you've talked again about them a little bit, but would you be able to elaborate on sort of how they work and how genomic testing helps that further?
Yeah, absolutely. So there's been an evolution of systemic cancer therapy. So we use very often what we call a chemo or cytotoxic chemotherapy, which are agents aiming to destroy cancer or rapidly dividing cells that these drugs are interfering with the cell division. So when the cell is divided into two, these medications intersperse between the DNA spindle and basically messes up the whole process of the cell division, and the cells die. Sometimes it also affects fast-growing cells, such as the hair cells or the bone marrow cells. So that's why you see anemia and decreased blood counts in patients who receive chemo, and also you see hair loss in those patients. Eventually, all these are transient, right? It goes away when you stop the chemotherapy, but that's a reason for you to realize how these medications work. And there's so many of them. We use them in combination, and each one of them has a different point in the cell division or the cell cycle where they act against. So that's what we combine them in many cases. There's targeted therapies now, which are basically small molecules that are a refined product of understanding the biology of the cancer. So they are aimed to interfere with specific cell pathways that are necessary for growth of the cancer, and spread that by blocking genes or proteins. So, for example, a very clear example of this is as straightforward as breast cancer having endocrine markers. We're doing targeted therapy for those patients because we're not giving typically chemo initially. If the cancer is driven by estrogen or progesterone, we use anti-estrogen drugs or endocrine therapy, and that is targeted drug, right? We're targeting only the pathway that the cancer is driving upon. But we have learned even further in learning how to block, cell signaling pathways that are altered by the mutation. So as we were mentioning, those mutations, for example, an EGFR mutation in cancer or an FGFR fusion in breast or NTRK fusion in other tumor types, what they do is that they basically get the cell into autopilot to grow, like basically giving all the time fuel to the fire. There's no control, so the cells grow exponentially and they don't stop. If you know what is the specific key to that keyhole that turns off the cell from growing and kind of normalizes the process, because that's the only thing that is acting up, then you make successful targeted therapy. So that's the rationale for this. Typically, those pills are taken by mouth. For many of them, there's small molecules that finish in NIB, like sorafenib, trametinib, everything that is IB, like it means inhibitory molecule, basically. So all those drugs are aimed to do that. The sort of targeted drugs, which are also immunotherapy combination kind of a thing. So they're monoclonal antibodies that are targeting against the specific biomarker in the surface, like trastuzumab for breast cancer is an example of that expresses HER2, basically goes against the surface and kills that cancer in that area. Immunotherapy is the other modality. It's the most newer of the treatment options that we have, but it's been in studies for many years. It's basically aimed to boost the body's natural defenses to fight and destroy cancer cells. So typically cancer cells are smart, right? So they say, don't kill me. I'm legitimate. I live here. I'm part of you, but actually they're not good. They're a spy trying to kill everything, but they're smart about it. They don't want to be killed. So they express molecules on the surface, or they stop the immune system to acting, basically calling the police, don't kill me, go elsewhere. And the idea here is to awaken the immune system or train the immune system so we can recognize those cancer cells as foreign or as malignant, and then eradicate them as normally will happen. So those are all the kinds of treatment options that we can do. And now there's a combination of them, all based on biomarkers, mechanism of action. So the field is very rich right now in combinations.
Honestly, pretty amazing to learn about, to hear all the new technology. We have a question wondering if you can use your insurance to pay for genomic testing.
So yes, most of genomic testing that I mentioned is covered by Medicare and by most insurance companies, particularly for advanced cancers. So, and a number of the tests are also FDA approved. So they basically have all the regulatory approval for this. Make sure that you sign up for the patient assistance program, because all of them have like forms that you fill and sign on the back of it. So they enroll you immediately in the assistance program, where they make sure that you don't have any surprise billing. I had experience with friends and patients that had gone through the process. Typically, if there's any issue with insurance, they will call you before they process the panel, and they give you the assistance that you need. Typically, anything that goes about $100. So at Massive Bio, I know for sure that our advocates are pretty good and helping patients navigate those waters too, particularly as we're looking for clinical trial opportunities for enrollment. So feel free to share that, and if you need any facilitation also, please free to reach out to them. They're pretty good at that.
Thank you. And for our final question, how do clinical trials relate to genomic testing?
Right. So, clinical trials, as I mentioned, they are based on new things that are happening in the space. So just to give you an example, just real quick, there's some evidence that survival could be longer if you actually match patients based on biomarker studies, right? So more data to follow on this. But just to give you, an example of an exceptional responder, is someone that I treated 7 years ago. It was a patient who had a tumor that is called cholangiocarcinoma, which is a rare tumor, but they tend to have different mutations in low frequency, but but in multiple of them. So they have BRAF mutations, they have IDH mutations, they have FGFR mutations. So all of them could be targeted within drugs. At that time, we had this drug that it was targeting BRAF, but it was used only in melanoma. And we actually gave the patient through an assistance program, basically a compassionate use program, combination of the treatment that it was for melanoma, and basically had almost an immediate response. And that led to a clinical trial that now has been published showing the same efficacy data that we found in a single patient, right? So the studies are helping other patients and yourself to get access to these drugs in an earlier way. So there's a lot of different phase studies. There's phase 1, 2, 3 clinical trials. There's CAR T cell trials, for example, where they target, where they extract your cells, train them and against the cancer itself, and then infuse them back again after that they're being grown. They have modified the CAR T cells to make them very advanced as well. There's the new generations coming, and those are all under clinical trials. There's CRISPR-Cas gene editing that was a subject of a Nobel Prize not too long ago, and now there's trials looking for that, doing changes on specific cells to either train your immune system to attack the cancer more efficiently or to modify disease. So all these have been leading to advances in information that we've been doing. So in pharmaceuticals, medtech, AI, which Massive Bio uses to match patients to studies, real-world data, consumer health, and that's why Massive Bio was created. It was created to use artificial intelligence to help patients translate the genomic testing results, and there's all these different tests available, but we need to scale it up, use technology for that, and that's what we're good at. So this is just a review reports you can see of Garden 360 and Foundation, all of them pretty comprehensive. We need to help patients navigate this, and we use it with deep learning technologies, basically. So the idea here is to find a patient that's the right patient at the right trial, the right location, the right time, and use technology for that. So genomic testing can lead to a couple of opportunities for trials. One is an umbrella trial, which is basically testing the impact of different drugs in a single type of cancer. So for example, lung cancer with 7 different biomarkers, you have 7 different drugs, but only for lung cancer. That's an umbrella trial. There's the basket trial, where they test the effect of drugs on a single mutation in different tumor types. So it doesn't matter where the cancer came from, as long as the cancer has that mutation, you can use the drug. So that's the TAPER, the NCA match study, and the My Pathway, Tapestry trials. All those are basically large basket trials. This is an example of the NCI match, where patients get genetic sequencing or genomic testing. They find the actionable mutation, they put the patient on the drug. If the patient response continues, and if progression, you test again. And if the patient matches to one of the new arms, then you continue on treatment. So really amazing how we are using the genomic testing and clinical trials to help patients to match to different options. This is just ASCO TAPER, for example. This is data, actually this is from quite a while now, 2014, but the idea is looking at all the different genes that are present in alterations and try to find at least one plausible genetic alteration that we can target. So that's the way of the future. And at CTCA, we have been using ASCO TAPER, enrolling our patients in that clinical trial, and we also have Tapestry now, so where we can find patients based on genomic alterations and try to see if we can help them to get access to those drugs earlier and helping other patients in the future for that purpose.
Thank you so much. I want to let the audience know, if you have any more questions about entering a clinical trial, about genomic testing, please click the link at the top of the chat, reach out to us on Facebook, head to our website. We are happy to provide you with any information you could want. Dr. Bonilla, before we head out, we have one more audience question. They're wondering, do you think removing positive margins by scar surgery ends a tumor?
Okay, so that's not a biomarker question, but based on my understanding of the question, is so it's can you say it again just to make sure that I'm getting
Do
it right?
you think removing positive margins by scar surgery ends a tumor?
Well, so I mean that's a surgical question, but typically when you are removing cancer, you want to leave nothing behind, right? So you want to went to the procedure itself. You want to remove as much as you can from the cancer and leaving free margins. Free margins means that when the microscope is being used to look at the edges of the sample, you see no cancer there. You only see a little bit of a scar or normal tissue that is at the edges. That means there was nothing left behind, at least to the best eyes of the surgeon. So we always encourage what we call an R0 complete resection, and that's to prevent the cancer from coming back. So hopefully that helps with the question. Yeah.
Thank you. And thank you so much for your time today and for giving us a lot more insight on genomic testing. It was really informative and great, and we're so happy to have you here.
Yeah, absolutely. So feel free to reach out to every one of us for anything you need at CTCA. Happy to see my patients here. If you want to help find clinical trials, you can use the Synergy AI ASCO, the app that you have when you're looking for ASCO taper, and the NCI match, the tapestry studies of the world, or any other trial that you may be interested about. 100%, they're there to help you. They actually do matching for patients for free. So you have a biomarker found in genomic testing, and you want to understand what is behind it. 100% recommend it, because certainly something that you may take advantage from and help you to find the best options for your cancer. And with that, just wanted to show you all the different pathways that are involved in cancer. This is not even including the stroma or the things that are related to the immune system. So a lot of things that we need to decode right now, but very exciting time for us to really advance science and help patients to find the best options against their cancer. So with that, I appreciate everyone's time today.
Thank you again so much, Dr. Bonilla. And thank you everyone for attending today. It was a great session.
Okay, take care.